next speaker is Chelsea Zhu from the Wei Wang Lab, right?
Yes. Good morning, everyone. Thank you for coming, and thank you for giving me this opportunity to introduce our tool, RNA-Skim, which is a rapid method for RNA sequencing quantification. So I will start my presentation with a very brief introduction from a high-level point of view about RNA sequencing. RNA sequencing is a leading technology to quantify the expression of thousands of transcripts simultaneously. The technology starts by reading out the transcript sequence into millions of short reads. And in order to reestablish the origin of this read, many of the data analysis starts by aligning this read into a reference genome or a reference transcriptome. And after the alignment, these aligned reads are assembled to determine and quantify basically these aligned reads are later being assembled to determine the presented transcript in the sample and also proceed for the quantification step. And the traditional pipeline usually includes using a top hat as an aligner coupled with cufflinks as an assembler, or using bow tie together with RSEM or Express for assembly. And as we can see that most of this pipeline require a lot of computational resource and time. If we want to analyze a reasonable size of data, usually it can take days, even with parallel computing. So to estimate the abundance of transcript, most of the alignment-based methods start by aligning the reads back to their origin transcripts. And this diagram here illustrates the reads that being aligned to a single transcript. The x-axis here shows the transcript location for the transcript, and the y-axis shows basically the read depth across different locations. And the abundance of the specific transcript can be estimated through the area covered by these reads. So in another word, the information that we're looking for, the sufficient information we're looking for is the area under the curve. And many existing methods, including the alignment-based or alignment-free methods, they try to integrate the area by calculating the height for every single base pair of this transcript. And since the curve is actually smooth, we only need a subset of this pair, and just use the height of this location to approximate the area. And in order to do so, we use this K-mer-based idea for the approximation. This K-mer ideas come from based on this given a transcript sequence. The K-mer are the consecutive and short subsequence of this transcript. And from a data mining point of view, this K-mer can behave as a feature of the sequence. And what we only need is only a subset of this K-mer to do the abundance quantification for speeding up the abundance quantifications. So based on this idea, what we propose is a novel type of K-mer named sigmers. And the definition of sigmers depends on the partition of the transcriptome. It is basically a special type of K-mer that occurs only in one cluster. And here's an example. In another word, these sigmers actually describe the signature of a cluster. So in this example is a transcriptome containing 5 different transcripts. And these 5 different transcripts are being clustered into two non-overlapping clusters. Cluster 1 containing 3 transcripts, and cluster 2 contain only 2 transcripts. And as an example of sigmer, GGAA here is labeled in the brown color. GGAA only appears in the sequence in cluster 1, but does not show up in any of the sequence in cluster 2. So GGAA here, we call it as a sigmer for cluster 1. And similarly, ATCG from cluster 2, which is labeled in orange here, it only appear in the sequence in cluster 2. So this we call ATCG as a sigmer for cluster 2. And sigmer can also be viewed as a special type of special reads. When we generate the reads from the transcript sequence, we can see from this example that only a sum of those reads contain the sigmers. And the reads that contain the sigmers are what we are interested in. So we use those reads to count the occurrence of the sigmer in the RNA-Seq data. And this kind of information are being further used to infer the abundance of different transcripts. So in another word, we basically use a sigmer to sampling a subset of the reads from the RNA sequencing data. And this type of information are being further used for the quantification. So this whole sigmer idea is implemented in the RNA-Skim. And here's an overview of the workflow, where we started from giving a transcriptome, and this transcriptome is first being clustered into a set of non-overlapping cluster based on sequence similarity. And the next step is the algorithm. Next step, we use the Bloom filter algorithm to discover all the sigmers for different clusters. And given the RNA-Seq data, the next step is to detect all the sigmer in this reads data. And we use a rolling hash algorithm to optimize these steps. And the occurrence of this sigmer are then used in the quantification stage. And in the quantification stage, the transcript proportion are estimated by maximizing the likelihood of observing all sigmers in the RNA-Seq data. So in the quantification stage, the maximum likelihood are being conducted through the EM algorithms. And in order to evaluate our method, we use the real data to evaluate the abundance reported by different methods. And the data comes from 56 inbred of F1 mice from 3 different cross strains. And since we do not know the ground truth, we compare the correlation with the microarray data. Since microarray only reports the gene expression instead of the abundance at the transcript level, so in order to conduct a fair analysis, we basically sum up all the transcript abundance for one gene. So basically, we estimate the gene abundance by summing up all the transcript in that given gene. And the result shows that, so we use Pearson correlation and also rank correlation to evaluate the methods comparing to the result reported by microarray. And the result shows that RNA-Skim is comparable to any of the methods with a very high correlations. And we also look at the running time on this real data set, which contain 44 million of parent reads. And the running time shows that the alignment-free methods, including the RNA-Skim and selfish, they're a lot faster than the alignment-based methods. And RNA-Skim is even faster, 10 times faster than selfish, because we only use a subset of the k-mer and a subset of the reads for the evaluation. So what we learned so far, the conclusion, is that the k-mer-based quantification is able to achieve a high accuracy in transcriptome quantification with a fraction of time used by alignment-based methods. And what we also learned is that not all the k-mers are needed in order to achieve the accurate quantification. And the software is actually available on GitHub to play around. And just to go over, since we are doing a tool, introducing the tool, so I just want to go over a few steps of how to run the tool. So the description is actually pretty it's pretty self-defined on the GitHub README page. But in order to run the method, basically it contains 5 different steps. So the first step is clustering, as I described, that you basically cluster all the transcriptome partition the transcriptome into different clusters. And the second step is discovering and also indexing the sigmer for different cluster. And these two steps, we only need to run it once, since if we're running using the same organism and using the same transcriptome, we only need to run this two-step once. And once we have the data, we will run step 3, step 4, and step 5. Step 3 basically, what I meant is step 1, 2, 3 is we only need to do it once for a given transcriptome. And step 4 for the counting step and estimation, we run it when the data is ready. So the counting step basically count the number of sigma, the occurrence of the sigma in the data, and the estimation step use the EM algorithm for the abundance quantification. And there are several parameters that we can play around. One is specifying the K, because we usually don't know what's a good K. So by default, the K is actually 60. But if the data is different, we can also play around with the K by specifying the RS length here. And the software also allows you to run on parallel threads, which is specified by the parameter of number of threads. So with that, I would like to thank Professor Wei Wang, who is the supervisor of this whole project, and a former colleague, Xiaojun Zhang, who developed this software, and everyone in our lab over here, and also the funding agency from NIH and NSF. And with that, I would like to take questions. Thank you.